Your eyes are a biological dashboard, a complex map of your DNA.
They were supposed to be identical. Same face, same freckles, in the same places. Then the eye doctor noticed. One twin had brown, the other had green. Not a trick of light. Two irises, sitting in the same genetic head. Meeting the world in two different colors. For most of the 20th century, the textbook story had been simple. Brown was dominant. Blue was recessive. Eye color was a single gene. Anything else, green, hazel, the in-between shades with no clean name, was a curiosity, a footnote. Then the genetics arrived. Researchers studying twins, families, and large populations began to notice something the textbook could not explain. Eye color was not one gene. It was not, in any clean biological sense, what high school biology had promised. Two genetically identical siblings could carry different eye colors. Identical twins could carry different eye colors. And green, the rarest natural color of the human eye, held by roughly 2% of the global population, sat right at the center of the confusion. Almost no one had it. Almost everyone had an opinion about it. Almost no one understood where it came from. The old story felt true because it explained so much in so few words. Open any introductory biology textbook, and you will find a Punnett square. The allele for brown, capital B, dominates the allele for blue, lowercase b. Eye color became a kind of folk genealogy. A small flag carried an iris. It felt like law. Family pedigrees did show patterns. But law in biology is rarely as clean as it looks on the page. The cracks appeared quietly. Brown-eyed parents with a green-eyed child. Blue-eyed parents with a hazel-eyed child. Identical twins who should have shared every visible trait, sharing almost all of them, except the color of their eyes. Then came the modern genetics. Genome-wide association studies. Hundreds of thousands of individuals. Millions of markers. The same machinery used to map disease risk and ancestry was finally aimed at one of the oldest questions in the family photo. What it found was almost embarrassing for the old story. The first major hits were not on one gene, but on a region, a locus on chromosome 15, near a gene called OCA2, and the gene beside it called HERC2. A single change in the DNA near HERC2, a SNP known as RS129-13832, was strongly tied to the difference between brown and blue. But that was only the loudest signal. There were more. SLC24A4, TIR, SLC45A2, IRF4, a long list of additional regions, each contributing a small effect, each nudging the color of the iris. The textbook had said one gene. The data said a dozen or more. Twin studies from the same era confirmed a related truth. Eye color is highly heritable, more than 85%, but never 100%. And then there was the color itself. Green, not a pigment. Under a microscope, a green iris contains no green pigment at all. The color comes from low to moderate melanin in the front layer of the iris, combined with the way light scatters as it passes through the stroma. The same physics that makes the sky appear blue, a trick of atmosphere rather than a true pigment, a color that exists in the meeting between fiber, light, and a precise absence of pigment elsewhere. And the surprise was bigger than the optics. Once the map was opened, it refused to behave like a single story. Light eye color clustered heaviest in certain regions of Europe and Western Eurasia, Ireland, Scotland, the Baltic Rim, parts of Scandinavia. But it appeared in smaller frequencies across the Iranian plateau, through parts of Central Asia, among Berber populations of North Africa, among the Pamiris of the high mountains. And the underlying genetic signal was often different in those regions. Different combinations of variants, different proportions. A light-eyed person in northern Spain and a light-eyed person in Afghanistan were often reaching a light iris by somewhat different genetic routes. That meant the trait was not the product of one origin. It was the product of many. Three frameworks began to take shape. The first was ancient. Light eye alleles appear to have existed among European hunter-gatherers long before farming arrived. Their descendants, the Mesolithic peoples of the late Ice Age, may have carried variants for light irises even though their skin was dark. There is no reason, biologically, why the pigmentation of the iris and the pigmentation of the skin must rise and fall together. They are partly separate stories, written on partly separate genes. The second framework came from Bronze Age movement, when farming populations expanded into Europe, and later when steppe-derived pastoralists moved in from the east, they met older inhabitants who already carried some of those lighter eye alleles. Admixture shook the variants into new combinations. Selection, possibly sexual, possibly only a relaxation of the strong pressure that favored dark pigmentation in bright sunlight, allowed the lighter variants to rise. The third framework was simple polygenic accumulation. Eye color may not have one origin. It may have many. Each contributing variant arose somewhere at some point on its own. They met, sometimes, in the same individual. Each framework held part of the truth. The most important turning point was a wave of large genome-wide studies published in the 2010s. By combining data from massive multi-ethnic cohorts, researchers confirmed dozens of loci for eye color, far more than a handful the textbook had suggested. Some of the newly discovered genes were not classical pigmentation genes at all. They were structural, developmental, regulatory, and the genetics of green turned out to live in the middle of this complexity. Green is intermediate on the spectrum from dark brown to light blue, the place where melanin levels are low enough to scatter light, but not so low that the iris washes out to clear blue. Variants in HERC2 and OCA2, working together with weaker modifiers elsewhere in the genome, push the iris into that narrow range. The color, in other words, is not produced. It is engineered, a small, delicate adjustment of many variables at once. That is why identical twins can sometimes differ. The genome carries the same recipe, but small, stochastic events in development, which genes it turn up or down, by how much, in which cell or which layer of the iris, can produce subtly different outcomes. But here is where the story turned in on itself. When researchers sequenced the DNA of a Mesolithic skeleton from Gough's Cave in England, a man now called Cheddar Man, who lived around 10,000 years ago, his reconstructed appearance was a quiet surprise. Dark skin, curly hair, and blue or blue-green eyes. Light colors in the iris, in a person whose pigmentation in the skin was anything but light. The textbook narrative had quietly assumed the two would travel together. Brown skin, brown eyes. Light skin, light eyes. The same latitude. The same evolutionary pressure. Cheddar Man undid that. Light iris color, the data suggested, was older than light skin color, older by many thousands of years. It had been moving through European populations long before the alleles that lightened skin had become common. Green, sitting between them, was not a midpoint between two recent inventions. It was a surviving echo of something much older. Picture, for a moment, the western edge of Europe in late autumn, the outer Hebrides, or the long Atlantic coast of Ireland. The peat bogs have darkened their veins in the soil over thousands of years. The grass is the color of old rope. The horizon is so wide the eye forgets what it wandered in here looking for. It is in places like this that green eyes cluster most densely today, among populations whose ancestors lived here for many thousands of years. Standing in that landscape, the genome feels less like a textbook diagram and more like a coastline shaped by tides. This is where the larger idea presses in. If I color, a single visible, measurable, almost playful human trait turns out to be older, messier, and more layered than the textbook claimed, then the ways we have historically tried to read identity from any single visible signal are too simple. Identical twins can carry different irises. Light eyes can coexist with dark skin. Green eyes can cluster in Europe and also emerge in Central Asia. Two people with blue eyes in two different countries may share little of their deeper ancestry. None of this proves any single political claim, and the science does not collapse into any convenient narrative. What it does is something more durable. It dismantles the idea that a glance is a genealogy. It reminds us that the human genome is not a flag. It is a long conversation with many voices, speaking across many generations, in a language we are still learning to read. Skin color and eye color tell us very different stories. None of them, alone, is a map. We will probably never fully close this version of the mystery. New pigmentation variants are still being discovered. Old ideas about single gene inheritance are still being quietly retired. But the science has done something more important than solve the mystery. It has enlarged it. The green eye, when we look closely, is not a single inheritance. It is a visible edge of a long conversation between many inheritances, many latitudes, many quiet meetings between lineages. It is older than the legends we attach to it, rarer than the stories claim. The map was never as simple as we drew it. The genome remembered things the page forgot. I do not know where the next green-eyed I will lead, but I know it will lead somewhere.